don't know is I actually graduated from this university also, but I couldn't make it to the university part. I was a kindergarten student here, so much older than all of you. In terms of experience, maybe, maybe one or two people. But so thank you for having me here. And so today I want to tell you about the recent experiments that we've been doing in my lab. I'm very excited about this project. It has not been published yet. Parts of it we submitted and are about to be published, but it took me a long time to convince the students in my lab that this is an interesting issue. So I hope it's interesting for you to understand what this structure means in a minute. So in immunology, as Arzu said, I've been in immunology, the self versus non-self discrimination is an important issue because our immune system should attack foreign invaders of the body, but at the same time should not attack our own self cells. And the idea in this project is that maybe something similar is happening at the molecular scale. So proteins like the ones you see here are perhaps making homodimers with themselves, but if they have close homologs, perhaps they have a choice, a molecular choice of making a homodimer as opposed to a heterodimer, and perhaps that kind of molecular choice allows the cell to integrate information. So this is we're trying to explore this kind of molecular choice. And so the focus is a class of transcription factor proteins, and in the human genome we have 49 of these. These are called BTB zinc finger molecules. As they're transcription factors, they have a C-terminal DNA binding domain that's composed of zinc finger motifs, and they interact with different DNA targets. And in their N-terminus, they have this other domain called the BTB domain. The name, unfortunately, was given by Drosophila people, and it has terrible long things that I don't know about. So I call them, I think it's Brickaback, Tramtrack, Broad, Complex family members. But anyway, we have 49 of these proteins in our genome. I guess Drosophila has quite a large number of them as well. And this domain is important because it allows these proteins to interact with each other and also to make homodimers. And on top of that, when a homodimer forms, other proteins can sit on top of the homodimer. And we'll see some structures that really show that. And what this kind of family picture shows is that every single family member on its N-terminus has this red BTB domain. So homodimerization or heterodimerization must be important. But in the C-terminus, these zinc fingers, they have different numbers and the spacings are different, so their DNA specificity must be different, but we really don't know what the DNA specificity of these proteins are. So for many years, we've been working on one of the family members called POTS1, another name is MAZ-R. This was first identified in lymphocytes. This is why I got into studying this field, because we were interested in immunology back then, and when you make a knockout of this gene in mice, it's very easy to screen the mice. You don't have to do PCR. The homozygous knockouts are, the newborns at least, are about half the size in terms of body size and in terms of weight, so you basically pick the knockouts out of the cage. So that implies that there is some kind of important regulation of proliferation during early development, and in fact we took mouse embryo fibroblasts from these knockout mice and compared them to wild-type mice and did some RNA sequencing experiments, and the most significant differences are in cell proliferation, but also cell cycle regulation and DNA damage responses. So that in that study we realized that this protein has some important functions in T lymphocyte development and T lymphocyte function, but also in early development and in general in a ubiquitous fashion has important functions in more basic things like proliferation. So more recently we've been making knockout models of this POTS1 protein, So in this case, we're using this human colon cancer cell line, which is very commonly used. And using CRISPR-Cas9 technology, we made a PAS1 knockout cell line and isolated RNA and did RNA sequencing. And we identify by differential gene expression 156 target genes. Of course, these target genes may not be direct targets of PAS1, so PAS1 may not directly bind to the promoters of these. It may be secondary effects. But of these 156, roughly half are upregulated and another half are downregulated, which means that this protein is, in some cases, depending on the context, is an activator of transcription, and in other cases is a suppressor of transcription. So to identify the real targets, we were lucky that there's this ChIP-seq data set in GenBank of PAS1 binding genes. So when we take the cross-section of the ChIP-seq data and our RNA sequencing data, we identify 15 target genes. So these we think are the, at least in this cell line, are the genes that PAS1 binds to its promoters and directly controls. So this is the list, and of this list, so 3 are upregulated when we remove PAS1, and the rest of them are downregulated. So just to show that this is really true, the top upregulated and downregulated genes, these we did qPCR, and in fact this gene called CTH1, which is involved in metabolism, is upregulated, and another transcription factor called ETV1 is dramatically downregulated when we remove PAS1. So ETV1 will be important in just a second. Keep that name in mind, in your minds. So another reason why we study PAS1 is recently this subclass of sarcoma cancers called Ewing sarcoma. People have been discovering in mutants, in patients, translocations between this common EWSR1 gene and the PAS1 gene. In fact, these 2 genes are on the same chromosome, in human chromosome 22, and they're about 2 million base pairs apart, facing opposite orientations. And in these translocations, the EWSR1, the function is not really known, but it's a transcription activator, but doesn't have a DNA binding domain. It's an RNA binding protein, and this part, the N-terminus of the EWSR1, is an activation domain. And in these translocations, this activation domain is translocated with the PAS1 gene. And if you remember the structure of PAS1, the N-terminus is this interaction domain, the C-terminus is the DNA binding domain. So most likely what's happening is this activation domain comes together with the DNA binding domain of PAS1 and forms a new transcription factor that regulates transcription differently in the tumors. So that was, and you know, every year now new patients are being diagnosed with this kind of inversion in chromosome 22. But actually, in Ewing sarcoma, the reason this gene was identified, 80% of Ewing sarcoma patients, they don't have a EWSR1 PAS1 translocation, but instead they have EWSR1 FLY1 translocations. And FLY1 is also a transcription factor on chromosome 11, and something similar happens. The activation domain of this gene fuses with the DNA binding domain of FLY1. And in fact, another subset of Ewing sarcoma is a translocation of the same gene with ETV1. ETV1, the target of PAS1. So we think there's some kind of regulatory loop where PAS1 is controlling the ETV1 gene and somehow regulating tumor development. And also, but this is only in a subset of translocations, and in many translocations the advantage of the tumor cells is because of other translocations. So that, I think, is an important issue leading us to the function of this PAS1 protein, not only in normal cells, but also in cancer cells. And earlier, we had also linked PAS1 to cancer because Emre Deniz and Nazlı Keskin, 2 PhD students in my lab, had discovered that PAS1 interacts with P53, the canonical, the most famous tumor suppressor gene. And what we found was that it's interesting, this interaction is not through an activation domain or another protein-protein interaction domain, but a region within the DNA binding domain. So this is the structure of PAS1, that's the BTB domain in the N-terminus. These are the zinc fingers. Within zinc fingers 6 and 7 is the region that they bind. And we made a model of PATS1, so that's the PATS1 DNA binding domain, and there's this negatively charged region that we think makes a pocket that interacts with PTP. So in conclusion, in this part, what I've told you is that this PAT1 I think is really worth studying. We've dedicated about 10 years of our lab here for this protein. It's important for gene regulation. Now we realize that it's important for translocations in sarcomas, but in general it has some kind of regulatory role in proliferation. There are some studies which we've duplicated that show that PATS1, knockout of PATS1 increases iPS generation frequency. So somehow proliferation of stem cells and cancer stem cells may be controlled by this protein. So to really understand how this protein is working, we wanted to do structure-function experiments. And to do that, we focused on this N-terminal BTB domain. And as I said, this is the protein-protein interaction domain. It can homodimerize or possibly heterodimerize using this domain. So this is a list of a family picture of the zinc finger BTB proteins. This is just a BTB domain, and this is the 49 human family members. And you see that there's quite a lot of conservation. The colors indicate conserved positions. There's one thing that's striking, and that's this one here, that happens to be PATS1. So compared to all 49 other family members, only PATS1 has this extra insertion in the middle of this domain. We don't know what that domain does. We wanted to figure this out. So to understand this, Sofia Piapelli, who's a PhD student in my lab, decided to crystallize this protein. So initially we wanted to crystallize the whole thing, but it turns out crystallizing DNA binding domains is very difficult because they're sticky when you express them, and in bacteria they stick to all over the place. So we just focused on this BTB domain. So Sofia purified this by size exclusion chromatography to purity, and crystallized the proteins. This is the first crystallization project in my lab. And so we got these different crystals, and in collaboration with Erika Mancini, who's a professor at Sussex University in England, who has access to the Oxford Diamond synchrotron, we diffracted these crystals. So this symbolizes our proteins in the crystal lattice. As you know, X-ray crystallography works by shining X-ray lights and collecting the diffracted light and identifying atomic resolution maps of the protein. So this is the crystal structure of the human PATS1 protein. And like other BTB domains that have been crystallized, it forms a homodimer. So blue and green are two different homodimers. They're identical to each other. They're mirror images. And in the middle here, these two alpha helices form this interaction surface that allows the dimerization. Unfortunately, there's this central loop that's missing in the crystal structure, most likely because this region is flexible in the crystal, and they don't, those amino acids don't really diffract to a good enough resolution, so we can't identify. And unfortunately, that's the region that's unique to PATS1, as opposed to all the other 49 family members. So what we were interested in, we couldn't really find yet. Turns out, this is the PATS1 protein sequence, the BTB domain of PATS1 protein sequence, among all vertebrates. And what you see is this middle region, which is unique to PATS1, is present only in mammals and birds, but is missing in fish. And when we heard fish, we thought of Stefan. And so we got some genomic DNA from Stefan, cloned the BTB domain of zebrafish PATS1, again expressed it in bacteria, purified it, crystallized it, and diffracted and got the structure. So now this is the zebrafish BTB crystal structure. Because this loop region is missing in the zebrafish, that part was less flexible, and we could actually solve the top part of the structure. But if you look at if you compare the two structures, they're quite similar. So the difference between the atomic positions are quite similar between two proteins. So using that information, Sofia was able to model this missing region in the human protein, And this, although there are some loops here, we can assign some new structures like these beta strands that form in the human. So this is the paper we just submitted, and hopefully will be published in the next couple of weeks. So when we look at the zebrafish structure, because it's an intact protein, we can do molecular dynamics. And Sophia did this, and you see is the interesting part of the protein is this top part. So in the top part, it looks like there's this gate. It's opening and closing. This is a 200 nanosecond simulation, and the protein has an open and closed forms. And I always imagine that there's maybe another alpha helix, another protein that gets stuck in between these two gates, and that's how the protein interacts with other proteins. But we don't know yet. So what I've told you, what I've shown you already is this is the human PATS1, this is the zebrafish PATS1. These are all the other PATS1, other BTB domains which have crystal structures in the database. So you see that the structures are quite similar actually. They form these homodimers, and they're very stable. So the dimers move with respect to each other, but they don't fall apart. And in fact, when you express them in bacteria or in human cells, they're almost always forming homodimers. Now, when we look at this family of proteins, this protein called BCL6 is the most studied family member. BCL6 stands for B-cell lymphoma, and it's upregulated in diffuse B-cell lymphomas. And BCL6 is a drug target and is interacting with core repressor proteins called SMART and core or B-core, which recruit histone deacetylases. In fact, BCL6, there's a crystal structure, and there's also a crystal structure of BCL6 bound to its core repressor. So the yellow peptides are coming from the core repressor, and they have this dimer on top of dimer kind of interaction with the core repressor. And a lot of B-cell lymphoma drugs are targeting this interaction, trying to separate the core repressor from the BCL6 BTB domain to try to kill B lymphoma cells which are addicted to this antigen. So we wanted to see if PATS1 can also interact with these core repressors, because as you saw, the structures are quite similar. So that's our mouse PATS1 protein. And when we just overlay the PATS1 and the BCL6 crystal structures, they look quite similar. I mean, the RMSD is quite low. And here is the BCL6 bound to the core repressor, one of the core repressors. And we predict that in this overlay, so this is a complicated overlay, but we have PATS1 dimer, BCL6 dimer, and the core repressor dimer on top of each other. And it looks like the core repressors could find some region in PATS1 that it can interact with. So this is our prediction of PATS1's interaction with the core repressor, shown in yellow. The PATS1 is green and blue. But in fact, that doesn't seem to be we don't think that's the case. So on the left here, you see molecular dynamics of the BCL6 interacting with the SMART peptide. That's the molecular dynamics of a crystal structure, of a peptide bound to the protein. Here, we docked the same peptide to the PATS1 structure. This is the modeled human structure, so you don't have the missing loops. And you see that even though the surfaces look similar in the two proteins, the peptide moves or in the dynamics runs away from the protein. So we don't think PATS1 interacts with these core repressors in the same way that BCL6 can do. Now there's evidence, and I'll show you later, that there is evidence that PATS1 interacts with core repressors and core, B-core, and SMART, but we think not in the same way, not using the same surfaces that BCL6 is using. So crystallizing the protein allowed us to really make hypotheses about how these proteins are interacting with each other. So this is a surface representation of these two proteins. On the left you have the BCL6, and how BCL6 binds to this core repressor is when the dimer forms, so you see the dimer in red and blue, when the dimer forms there's this lateral groove. I don't know if you can see it. This is the side view and that's the bottom view. the surface generated by the dimer makes this groove that the peptide can sit on top of. And when we look at our crystal structure, in fact, there is a lateral groove on the side here and also on the bottom where the dimers are forming, but our dynamics indicate that it's not this lateral groove that is used to bind to the co-repressors. And perhaps it's the top part of the protein where you saw this opening and closed, and that's the model that we're working with right now. So, in conclusion, in this part, I've shown you that this 49 family members of the BTB domain transcription factors, they make these stable homodimers, and the homodimers make this interaction interface that allow their interaction with other proteins. The obvious questions are, so what controls this dimerization? Is it like a jigsaw puzzle where the two proteins that are identical fit, or are there some sort of amino acids that are critical for these interactions? Now, with the crystal structure, we can address these issues. So these are non-covalent dimers. So, and we have 49. And in my fantasy, there is this 49 by 49 matrix where we can test how the proteins choose their homodimer partners as opposed to heterodimer partners. If the proteins are so similar, why do they, or do they make homodimers preferentially over heterodimers? This may be important for gene regulation as well, because you can imagine they have different DNA binding domains, so they're going to bind to the promoters and enhancers of different genes, and a homodimer may bind to a certain sequence, but a heterodimer may bind to a different sequence, so there may be some kind of combinatorial specificity that controls gene regulation. So I'm really interested in finding this choice of homodimer versus heterodimers. And to address that, I would like to figure out the affinity of the homodimers to each other. So, to remind you again, we're dealing with this 49 by 49 matrix, or 49 different proteins. We know that some of them make homodimers because they are crystal structures. Can they make heterodimers? So to address this, we set up this in vivo, in cell system of interaction. So the system works as follows. We're using a baby hamster kidney cell line. This cell line has been modified so that in its genome there are 256 concatenarized binding sites for lac repressor, okay, the bacterial lac repressor that you know from your cell biology experiments. So the lac operator sequences. And in this cell line, we transfect 3 different plasmids. In the first plasmid, we express the lac repressor, so lac-I encodes the lac repressor, which is fused to a GFP binding protein, which is a nanobody. We'll talk about nanobodies in a couple of minutes. So this is a GFP binding protein that will recruit GFP. In the second plasmid, we express a tag GFP protein, which is a modified GFP, that's linked to a BTB domain of our choice. And in the third plasmid, we express tag RFP fused to either the same BTB or a different BTB. Okay? So in this system, what we expect is we have these lac operators, 256 of them, and the lac operators bind to the lac repressor that brings this nanobody to this gene focus in the chromosome, and that recruits by the nanobody GFP interactions, recruits the first BTB domain, and if that BTB domain can form a homodimer or heterodimer, that would recruit RFP. And on top of that, adding a fourth plasmid, we can also put the co-repressor tail onto a blue fluorescent protein. So in the experiment, you'll be looking for green dots that are co-localized with red dots in the nucleus, that in some cases will be co-localized with blue dots. Okay? So there are these are concatenarized sites, and we don't know which chromosome has been inserted. We bought this cell line from a company, but then we made all these constructs ourselves. So the first type of thing that we can address with the system is, does BCL6 homodimerize, and can it interact with SMART rancor in the system? We know it does because there's crystal structures, but can we show can we use this system to show this? So in the system, we're expressing the Lac repressor GFP binding protein, which will recruit the GFP tag BCL6 BTB domain. If that's homodimerizing with its own self, then RFP will be recruited, and then on top of that, if that interacts with SMART or ANCHOR, then BFP will be recruited. Okay, is this system clear how it works? Okay. And this is the result. So, so this is the several cells. We've taken three different pictures with three different fluorescence cubes, and here you see the GFP expressing nucleus of a cell, and inside the nucleus there's this GFP focus. That's where the Lac repressor GBP recruits the protein. You see that there's GFP that's not in the focus because probably there's more GFP than the Lac repressor, so not everything is recruited to the focus. But in the red channel, you see that there's a lot of red in the cell, but there's a red focus. So that is dependent on the BTB homodimerization of BCL6. And on top of that, there's a blue dot there, which is the co-repressor binding on top of the BCL6 dimer. So I really like this because, I mean, to show that in a living cell, these are live cells, that three proteins are interacting. Showing a trimer is not that common in biology, I think. Hopefully we'll be able to show using infrared fluorescent protein, maybe we can show tetramers forming as well. Okay, so we've done this with many cells, and we always get a red dot, a green dot, red dot, and blue dot co-localizing. So now that was with SMART. This is with ANCHOR. We look at many other cells. So again, green dot here, red dot, blue dot. In some cells, the cells are about to divide, and so they've gone through S phase. So the cell hasn't divided yet, but it's duplicated as chromosomes. So we see two dots, but both dots have both red and blue co-localized. So the next question, obvious question, was, okay, so BCL6 can homodimerize. We knew that. It can bind to SMART or ANCHOR. We knew that. But can we show PUTS1 homodimerization? So green PUTS1 BTB, red PUTS1 BTB. And yes, so we have a green dot, a red dot. They're co-localized. We counted numerous cells. And next, can PUTS1 interact with these co-repressors? There's one paper that claims that they do with a not too clear experiment. So here we're expressing GFP tag PUTS1 and BFP tag SMART or ANCHOR, and in fact, so green and blue are co-localizing, so PUTS1 can interact with the co-repressor. We don't know, we don't think it interacts in the same mechanism as BCL6 is interacting, but it does interact. That's with SMART, and that's with ANCHOR. Again, you see green dot and blue dot co-localizing. Okay. Some other questions we've addressed. So is it necessary for the ANCHOR? So this is a control for the experiment. ANCHOR and SMART, without ANCHOR and SMART, just expressing BFP. So is there some affinity of BFP to the GFP, for example? And that doesn't seem to be the case. So in this case, we have the PUTS1 on the DNA. You see the green dots, but you don't see the blue dots co-localizing where you have the blue dots. So you really need this peptide. It's about a 10 amino acid peptide from SMART or ANCHOR that fits on top of the PUTS1 dimer. So then we started to build this matrix of this, 49 by 49 interaction matrix. So PUTS1 and PUTS2, which is another family member, can they form heterodimers? So here we have green PUTS1, red PUTS2, recruited to the DNA with the Lac repressor. And in fact, yes, so you see green dots, you see red dots, they co-localize. And, but that's not true for every BTB domain. So BCL6 and PUTS1, okay, so green PUTS1, red BCL6, you see the green dots being recruited to the DNA, but you don't see red dots forming on top of them. So PUTS1 is in the DNA, but BCL6 cannot heterodimerize with them. So as I said, we're building my fantasy matrix. So we have red tag proteins, green tag proteins, and we're one by one testing them. You can see in the diagonal are the homodimers, and with one exception, the homodimers can always form. The only heterodimer we've identified so far is the heterodimer of PUTS 1 and PUTS 2. So red PUTS 2, green PUTS 1, or green PUTS 2, red PUTS 1. We've done it both ways. So that somehow, even though the names are the same, the names are not because the proteins are similar. The identity between PUTS 1 and PUTS 2 is quite not too high. But so far that seems to be the only heterodimer that can form. And hopefully in the future we'll be able to fill that matrix. When I first mentioned to my students, they said they thought I was crazy. I don't even know what 49 by 49 is, but so we're slowly building it. And now I think now that they saw that some heterodimers can form, they're all excited. Everybody wants to clone a BTB domain. So how we think this is happening is as follows. So this is the crystal structure. So what I'm going to show you is at the molecular detail how these foci are forming in the cell nuclei. Okay. So this is the crystal structure of the Lac repressor DNA binding domain bound to the Lac operator sequence. In fact, it binds as a tetramer in bacterial systems. So we think it binds as a tetramer to our Lac operators in these baby hamster kidney cells as well. So this Lac repressor we express in the cells with a GFP binding protein fusion. So it's a fusion protein that has Lac repressor DNA binding domain and GBP. And that GBP recruits a green fluorescent protein. Okay. So the sizes of these are all crystal structure data. So we have 4 GBPs, and we envision 4 GBPs recruit 4 GFP molecules. Now the GFP molecules are expressed as fusions with our BTB domain. So in this case, this GFP is fused, let's say, with a PUTS 1 monomer. That may be in the focus. A PUTS 1 homodimer that has 2 GFP tagged PUTS 1s may be in the focus. If we express, let's say, a BCL6 homodimer, but one monomer is green and the other is red, so this is the red fluorescent protein. This is what you're seeing when you see the green and red cells, red dots co-localized. And also, I've shown you that they can be heterodimers. So in this example, green tagged PUTS 1 and red tagged PUTS 2 can heterodimerize. That's what you see when you see green and red co-localizing. And on top of that, when you see green, red, and blue co-localized, this is what's happening, we think. We have a homodimer. On the surface of the homodimer forms the interaction interface of the smart Arancor peptides, which are shown in these green residues, and that brings a blue fluorescent protein, 2 blue fluorescent proteins. Okay? And you have to imagine these sequences are repeated 256 times. So you have this structure, this kind of structure repeated multiple times. Clearly, you're not seeing a single GFP molecule in the nucleus. You're seeing multiple of those. We don't know how many GFPs we need to see in our system. What is the sensitivity of the system? But at least we can use this system to allow to address whether the proteins have some affinity to each other, and how they interact with third-party proteins. So in conclusion of this part, I've shown you that these BTB domains are really stable. When you express them in bacteria to crystallize, or if you express them in mammalian cells, they form these dimers, and they don't. We've done live cell imaging, and the green and red dots don't diffuse. Once you get the co-localization, it stays there for multiple hours. The homodimers we think are favored over heterodimers. We don't really know why this is. We could only identify one heterodimer. This is what I meant by sibling rivalry in the title of the talk. I think of these BTB domains as siblings, and because they're so similar to each other, but they don't like each other, it seems. And we've been using this F2H assay, fluorescent 2 hybrid assay, to model these dimerizations. Now, the obvious questions that need to be addressed is, what is this 49 by 49 matrix that controls this dimer universe? And what is the extent of heterodimerization? As I said, we only know of one so far. In the literature there are some, but we're using this system to test those. And moreover, can we control dimer choice? If you remember, BCL6 is this cancer oncogene that is targeted with drugs that try to prevent the dimer formation. So can we design drugs that will either force homodimers or heterodimers forming or break those apart? So to address this in the final part, I want to show some more recent experiments with this nanobody technology, which I'm very interested in lately. So nanobodies are the, artificial biotechnologically generated proteins. They are parts of antibodies that come from cameloids. So if you, on the left, you see our typical mammalian antibodies. We have heavy chain and light chain containing antibodies, and they're quite large in size, 150 kilodaltons, the normal size of an antibody produced from B lymphocytes. But in a subset of animals, so camels, cameloids, so camels, llamas, alpacas, and dromedaries, have this, in addition to the heavy chain, light chain antibody, they have a single heavy chain antibody. And just like the normal antibody, this heavy chain only antibody binds to the antigen using this variable region in the N-terminus. So what you can do is you can clone just the antibody binding part, and that makes a very small protein, 1/10 the size of a normal antibody, which has some advantages. First of all, it increases affinity because it can go into small antigen binding sites that a normal antibody cannot bind. And expression in heterologous systems becomes much more easier. You can express them in bacteria, which we did. And you can also, so when the technology first started, it was an experimental mistake that turned out to be that allowed a Belgian scientist to discover that these things exist only in camels. Also, some types of sharks also have this single domain antibody. So when the technology was first developed by a Belgian biotech company, they were injecting antigens into llamas. So you need a llama farm. And just as a aside, there is a llama farm in Turkey, in Konya. So there's this guy in the agriculture school who imported llamas from South America. I contacted him, but then it turns out you don't really have to inject the antigen to the llama. What you can do is you can do phage display. So you can make a naive library of your nanobodies. So make very random mutations in the CDR, complementary determining regions of the antibody, and then do selection using phage display in E. coli, which is much cheaper and faster than going to an animal farm. So we did this in collaboration with Ario De Marco, who's a professor in Slovenia, and so Sara Barakat, who's a PhD student in my lab, made an antibody against the PAX1 BTB domain using phage display, and we identified 49 clones by ELISA that have nanobodies that bind to PAX1, and we expressed them in E. coli, and just to visualize them, we tagged them in their C-terminus with the M-cherry fluorescent protein. And so out of the 41 clones, we have 6 of them that we think have high affinity. So this is a size exclusion column experiment. So we've purified the PAX1 BTB domain. We think it forms a dimer, and in the size exclusion, it migrates where a dimer should migrate, around 44 kilodaltons. We express also the nanobody fused to the M-cherry. Normally the nanobody is 15 kilodaltons, but when you tag on M-cherry, it becomes about 30 kilodalton protein. And if we incubate them 1-to-1, now we think there's this nanobody in yellow and the PAX1 BTB domain complex, and now you get about the 60 here in the black curve, you get the a higher order complex. So using this assay, we're testing the affinity of the nanobodies to the PATS1. Of course, this is a model, this is our crystal structure, and that's a model of the nanobody. We don't know if the nanobody binds to a surface on the dimer or whether it only recognizes a monomer. These are issues that we're working on right now. We can also envision, since these are small proteins, we can express them in cells. So in this system, we're using the same fluorescent 2 hybrid assay. So the light repressor tetramer, PATS1 monomer with a GAP, or PATS1 dimer with 2 GAPs. And we can express our nanobody tagged with cherry. And if the nanobody binds to its target, it should get colocalized, and in fact it does. So green PATS1, red nanobody, and they're co-localizing in this focus. Okay, so these 6 nanobodies, we're testing which one has the highest affinity. This is an experiment from last week, so quite recent results. So in conclusion, we're interested in these BTB domain structures. We've crystallized one, but hopefully we'll be able to get more crystal structures. This is an ongoing collaboration. They're conserved structures, but they have some choice between homodimers versus heterodimers. And they interact with these co-repressors, which recruit histone deacetylases that allow the suppressive transcription function of the proteins. And so far, we think homodimers are preferred over heterodimers, but we're addressing this issue. Hopefully, my matrix will address this issue. So I hope I've shown you some glimpse into how our studies and how the dimer choice is determined, and the outcome of this would be transcription regulation that's relevant to cancer, especially sarcomas. This is only one project, so I wanted to take a couple of minutes, if I have some more time, to discuss some other projects that are ongoing or will be starting. So I think one interesting thing is we're trying to get sarcoma cell lines that have this PATS1 EWRS translocation. Well, if we can find this, we can clone this translocation product. We can compare its activity to the normal PATS1 and try to find out what's driving the sarcoma. Now, this F2H assay is kind of like a yeast-2H assay, yeast-2 hybrid assay, but it allows us to do a high-content screening under the fluorescent microscope, trying to find drugs that will dissociate the green dot from the blue dot, or perhaps, start with 2 proteins that cannot heterodimerize, and then add a drug that will bind them together. So we plan high-content screenings under the fluorescent microscope, using small molecule libraries to design drugs targeting these dimers. And BCL6, as I told you, already has these kinds of small molecules, but the other family members, especially PATS1 in sarcoma, may be good candidates. And as I said, we know that these nanobodies bind to the PATS1. We'd like to crystallize the complex, perhaps identifying the real structure of this top part of the protein, which was flexible. And so nanobodies are typically used as crystallization chaperones in crystallization studies, and in this case, perhaps we could use that kind of system. Moreover, this complex, the green, and I think it wants me to stop. The complex, what I've shown you in that model crystals structures is move too much. So we know there is at least 3 proteins in the complex, but perhaps there's more. Perhaps forming a homodimer or a heterodimer and then having a co-repressor bind is recruiting other proteins to this gene focus. And perhaps a repressive chromosome domain is forming on the foci. And we can address this by sort of precipitating these foci and then trying to identify what's present there. Are there histone deacetylases? Are there activating proteins? Is there RNA polymerase? Where is the location of this protein inside the nucleus? Is it near the nuclear lamina? We don't really know these. The dots don't seem to be in the middle of the nucleus, but as you know, chromosomes have domains within the nucleus, and this chromosome where the focus is, how it's moving around inside the nucleus, and is it interacting with other nuclear compartments, is interesting outcomes of this system that we've set up. I think I would like to really build on this nanobody screening. In my fantasies, there's a nanobody company that allows us to maybe commercialize some nanobodies in Turkey. But the phage display technology is quite simple. All of that stuff that you've seen with the nanobodies, Sarah went to Slovenia for 2 weeks and did the screening in 2 weeks, and then took about 2 months in our lab to express and purify the protein in our lab. But the phage display technology allows you to, once you have the library, to make nanobodies against anything you want in the universe. And finally, there's this, I'm really excited about this 1004 project, which was just presented last week in TÜBİTAK, which is in collaboration with Ahmet Gül in Istanbul University. And in this project, we're trying to find alternative drugs to inflammatory pathways. So IL-1 receptor is his favorite subject, and we're proposing to make nanobodies that will be used as extracellular drugs targeting IL-1 receptor inflammatory diseases. And so for example, colchicine is a common drug that's used against these IL-1 receptor inflammasome diseases, and anakinra is another drug that's been developed, so we hope to make nanobody equivalents of these, which hopefully will have higher affinities and better function. So, so that, I think, is the future for me. So this work has been done with some collaborators. The molecular dynamics work has been done in collaboration with Canan Atılgan. This is Sofia Piepoli, who's an Italian citizen who came to Turkey as an Erasmus student and fell in love with Turkey too, but also a Turk, and got married, and now is Turkish kind of, and starts to speak Turkish. She's done all the molecular dynamics, the crystallization, and the protein expressions. That was in collaboration with Erika Mancini at Sussex University, and we used the Oxford diamond synchrotron for the diffraction of the crystals. The nanobody work we've done with Ario Da Marco, that's a TÜBİTAK bilateral project, which has 2 more years, so we hope to make many other nanobodies. Tolga Sütlü, when he was at Sabancı, was my closest collaborator. We have common interest in immunology. I'm not too interested in NK cells, but these BTB domains, they're family members, zinc finger BTB proteins that are critical for NK cell function. So I think there is some kind of possible collaboration in the future about that. Uygar Tazebay, as a close friend, we have a common project on another protein called CCDC 124. Maybe last time I was here I talked about that. This is the protein that localizes in the mid-body of the cells and has a function in cell division. And Safa and Ahmet are in Marmara University. We're collaborating with human diseases, genetically inherited diseases in Turkish populations. So the experiments were done mostly by Sofia, but also Melike, who did the RNA sequencing and the qPCRs. Line did all the fluorescent 2 hybrid assays. She unfortunately is going to Germany to do a PhD. She finished her master's studies. And Sarah was a dentist from Yemen and came to Turkey, also learned Turkish, and is now turning into a molecular biologist. And so this is Melike Gezen. Nazife started the F2H assays, and Hakan, they're both in Germany, and Sofia and Sarah are now working on these. And we had some help from TÜBİTAK for money. So thank you very much.
Should I have some questions?
About this 49 to 49 matrix, I'm sure you have thinking about it. But all these 49 BTBZ zinc finger proteins are expressed at all the cells at the same time, or
No, of
there are
course not.
some preferences?
Yeah, of course. I mean, I think there's a subset of them that are ubiquitously expressed. Many of them are tissue specific. I think a derivative of this question is like the crystal structure of the zebrafish that I showed. This is the question I get all the time. Like, does the zebrafish bind to the human? We haven't done that yet, actually. But I think it's not really physiological. So I think the matrix is not that big. So
Yes.
you know, we have to find the ones that are expressed in the same cell. But the ones I've shown you are expressed. So BCL6, POTS1, and POTS2 are expressed in lymphocytes, for example.
Very nice
Right.
talk, very nice study. Thanks. Well, I was wondering, these proteins, are they monomers at the beginning? I mean, how they are, you know, as soon as they are produced, they become dimers.
Yes, that is something that's really perplexing. So there are several, you know, in our last molecular biology congress in ITU was this talk which was really eye-opening for me, which is why everybody should go to the congresses, even though it seems expensive and, you know, not on par with foreign molecular biology congresses. I think it is. So that after hearing this talk, and I forget the guy's name, he was a German guy talking about chaperones, famous, but I forget, I apologize. So how can a protein form a dimer? So these are cytoplasmically expressed, so they're expressed on a ribosome. So when you think of a ribosome, this guy was talking about polysomes. And so when a protein is coming out of a polyzome, the closest thing to it is the protein coming out of the next ribosome on the polysome, right? So you can understand co-translational dimerization. So that was an E. coli system, I think, where you know, polysome proteins can form co-translation. I mean, maybe that's something that's forcing this homodimer choice. Maybe homodimers are forming because all these proteins are expressed on polysomes. But that doesn't seem to be the case, because if we tag one protein with RFP and another protein with GFP, two different plasmids, so that's not a polysome, but you can see dimerization. So, you know, co-translational dimerization is not necessary to form homodimers. That's my conclusion of that. When a heterodimer is forming, clearly it's on different ribosomes that are being translated. But they're translated in the same region. They're, you know, in the cytoplasm somehow. But once they form, they're quite stable. Only one, the one that doesn't homodimerize in our system, BAH1, makes disulfide bonded homodimers. Okay, so we can't screen for that because in our system there's no disulfide bond. So I mean, I'm hand waving. I don't really know how a heterodimer or a homodimer forms and why it's stable. But we know when we express them in E. coli, the protein it comes out as a dimer. We freeze them, we thaw it, we do many things to it. We crystallize it, and it stays as a dimer. There's one example of a BTB that can form a tetramer. A tetramer is interesting because if you imagine a dimer binding to DNA and another dimer binding somewhere else in the DNA, that may explain this looping phenomenon, which is important for transcription. Right? So, in fact, when you look at if you remember the first slide, the ChIP-seq data, the POTS1 binding sites in those 15 targets, they all come as two binding sites. So you have a dimer binding here and a dimer binding here. So maybe POTS1 is doing this kind of tetramerization by looping. We don't know. We tried hard to show that in our crystals we could find tetramers by size exclusion, by small angle X-ray scattering, but we don't have evidence for tetramer formation. But out of the family, there's one that can form tetramers. Now the other thing is, of course, this is an artificial system. We're only looking at this BTB domain. The proteins actually there's this DNA binding domain, which puts it in a totally different context. Right, on the DNA it has this two-dimensional life, whereas in our BTB domains they're floating around in the nucleus or in the cytoplasm. So maybe we're doing an artificial screen for dimerization, but at least it will be a quick and dirty way of figuring out what can and what cannot do tetramers. I don't know if that answers your question.
Yeah, but I was like more expecting if there is a factor that keeps the proteins as monomer, so when it's needed it can dimerize or tetramerize.
Yeah,
or
yeah.
whatever. So is there any evidence showing that?
No, but again, in a hand-waving fashion, I can give this answer. If you look at the, I don't know if I can show you. If you look at the these green and red dots, let me find a good one. Okay, so here, I'll focus on this cell. Okay, so we're recruiting the green protein to the focus. Okay, so you see the green dot. The green protein has a nuclear localization signal. This is how we designed the system. Okay, so because normally the nuclear localization of the transcription factor is close to the DNA binding domain. Since we don't have that, we added a nuclear localization signal. So you don't see any cytoplasmic green protein. All the green protein is in the nucleus. And a subset of that green protein is in the focus. But the red protein, we didn't put a nuclear localization signal to make make it hard for the to prevent some kind of non-specific interaction. So all the red protein and you see in the cytoplasm there's quite a lot of, in all the cells there's a lot of red protein in the cytoplasm, but I think, it's just a hunch, all the red protein that you see, this is the one that does, oh no, it does interact here. So here you see all the protein in the nucleus is recruited there through its interaction with the green protein, I think. And of that, there's a subset that can interact with the green protein in the focus. Okay? I don't know if that kind of answers. So the protein interaction is strong, I think. So that, I don't know if the proteins are entering the nucleus through the nuclear pore as a dimer, or if there is a dimer in the cytoplasm, it dissociates, goes through the nuclear pore, forms a dimer. These are things that we can address, I think. Making mutations in importance or.
By the way, these percentages, the 60% or 50, 72%, 74%, these are percentages.
So the way we did the experiment is, we take many pictures. In the pictures, we count only the cells that have a green dot. Of those cells, what percentage of the cells have also a red dot co-localizing? So theoretically, it should be 100%.
Yeah.
But we unfortunately we don't have access to our confocal microscope for complicated reasons. So these are done on a fluorescent microscope, and the red dot may be in a different plane that we can't visualize. So that's why I think it's not 100%.
So thanks a lot. Very interesting systems, fluorescent one and the nanobody one too. My question is a little bit of a curiosity, right? So this BCL6, that's very famous protein in B-cell lymphomas and so on. And I remember from some past studies, it has some gene regulatory interaction with P53, which reminds me, since your PATS is interacting with P53, is there any evidence that BCL6 is interacting with P53? I don't know if there's any publications on that, or if have you tried BCL6
Yeah, let me try to
directly
remember.
interacting with P53 at all, or
So
that would be very interesting,
yeah,
I
I
think,
think so.
right? I mean, it would put.
There are many ZBTBs that are, there's a Korean group that works with these zinc finger BTBs, and they have this kind of two hybrid screen factory, and in that system they show BTB interactions with many of the family members. Okay. We were the first to show the PATS1 interaction, but many of those 49 also bind to P53. I can't remember if BCL6 is one of them, but what BCL6 is important for germinal center formation. So BCL6 knockout mouse, you have no germinal centers, no class switching of antibodies, you have low affinity antibodies. So it's important for T lymphocyte development and antibody generation. BCL6 in the germinal centers, so when antibodies are forming, there's this mutagenesis process, okay, somatic hypermutation. And there's DNA damage. And the cells should respond to DNA damage by upregulating BCL6 and upregulating P53. right? Because p53 is this canonical DNA damage sensor, right? When there's DNA damage, p53 and MDM2, this is another part of my studies which I didn't talk about here. MDM2 and p53 are normally interacting, and that causes degradation of p53. When you do DNA damage, or if you give drugs like Nutlin, you separate them, p53 accumulates after DNA damage. So in that model, in B cells that are undergoing mutation, the system doesn't want the B cells to undergo DNA damage response and die. So BCL6 is upregulated, it inhibits p53. So the cells don't give a DNA damage response during the somatic hypermutation pathway. That's a very specific interaction. But I think that's at the level of gene control. So BCL6
Yes,
is inhibiting the gene expression of p53, not
Exactly.
protein-protein
Exactly.
interaction. Yeah. But
That what made me think, since both are similar proteins, do they also interact? That would put another twist to the whole.
yeah.
Even in the germinal centers, or in
But the PAX1 is expressed in T lymph. It's expressed ubiquitously, actually, as you saw. The mice are have a phenotype early on. But most studies of PAX1 have been done on T lymphocytes, nothing on B lymphocytes. So that would be interesting.
I'm wondering whether PAS1 and PAS2 are topologically similar.
The reason they're both called PUTs is so the name. They have this BTB domain in the N terminus and a zinc finger domain in the C terminus, but only these two out of the whole family have another domain called an AT hook. That's the A in the PUTs. Other than that, the BTB domains are not similar. Okay? And if you look at the sequence similarity, I think they're about 60% identical or something.
I'm asking this because PAS1 dimer is an obligate complex structure, and it's when I look at it from structural point of view. So maybe it's not stable on its own, like as a monomer.
I think all of them. I mean, if you remember the dynamics of all the known crystal structures, once the dimer forms,
They just
they don't separate. I think this addresses Ibrahim's question. I mean, there's something in the structure that kind of like a jigsaw puzzle fits the homodimer. But why? How then can we see? I don't know if that's the picture. How can a PATS1 and PATS2 form?
that's why I asked that
Yeah.
maybe they are topologically similar, and so they can fit. I mean.
So Sophia has done modeling of a PATS1 monomer and a PATS2 monomer, docked them, and did the dynamics, and those are also stable.
Because there's some kind of spreading play, so there's.
Yes, there's a beta strand on the bottom that forms a beta sheet, a 3 beta sheet complex. On the bottom it keeps it together, but then there are those central alpha helices that make the major contacts. We talk about this in our crystal structure paper. I'll send you to.
In your 3-color experiments with the nanobodies, you also have the competitor or the substrate for them as well, the BTB domains, right?
Yes. So
And-
green is BTB, red is BTB, and blue is the core repressor.
Anchor, yeah. So, as a follow-up question to maybe Ibrahim Hocaz, I think the BTB domains would require to be dimerized to have a complex with them, right?
I think so too.
So would you maybe check if they are also triple positive in the cytoplasm, since they would have the
That would require, I mean, yeah, so we don't really know because the green is all in the nucleus. You don't see any green in the cytoplasm.
in these ones, right?
Yeah. In any green, all the greens have a nuclear localization signal.
Hmm.
I thought what you were going to ask was the following. So let me go to this model. So I show you the BTB, so blue fluorescent tagged co-repressor, the smart co-repressor, binding to a homodimer. Okay? But we don't really know this, of course. The co-repressor may be interacting with a monomer. But because of the crystal structure and this lateral group that forms, we think it's the dimer that binds to the co-repressor. Now another question that we would like to address is that can a heterodimer like this PATS1, PATS2 heterodimer, can that interact with And that would open up a lot of possibilities. Maybe homodimers are activating or repressive proteins because they recruit co-repressors, but heterodimers turn them into activated proteins, and that's why the gene targets there are some upregulate, some downregulate. So those are things we'd like to play with. I mean, this is kind of a model. So what we've been talking about is, I think this does not exist in these foci. I think once the protein is formed, it's as a dimer. And what I showed you in the PUTS1 is, if you remember, we had green and red, green and blue. We didn't have green, red, blue for PUTS1. For BCL6 we had green, red, blue, but for PUTS1 we had green PUTS1 and blue co-repressor. So clearly the co-repressor can bind, so a green that's docked to the focus using the nanobody recruits the complex, but another green homodimerize, and the smart peptide can build on top of that. Of course, this green may be bound to another nanobody in the complex. We don't know how. What is the stoichiometry of green protein to red protein, for example?
Thank you.
But I understand. So one curiosity is the structural aspect, right? Like to understand how that works at the structural level, how they interact and stuff. But you said you have chip-sec data for PUTS1.
Yes.
Can you not look around the binding sites for PUTS1 if you find other binding sites for any of the other ZBTBs that could actually make a biological interaction there?
The target site is not well defined. Zinc fingers like to bind to G-rich sequences. So when they do the PUTS1 chipsex, the way that experiment was done is it's a GAP tagged PUTS1 overexpressed in 293 cells. Actually, this is a Stanford group where they GAP tagged all transcription factors. So there's chipsec data for everything. But the target, so when we didn't do this, but we started in collaboration with Erman'ın. When you compare the targets, you can't really differentiate a PUTS1 target than like a BCL6 target. So I think finding, clearly they have different targets because they have different number of zinc fingers and different binding affinities and stuff.
But okay, then if they have chip-sec data for all of those 949, then you can look again for co-localization.
Yeah.
And that would narrow down your matrix of 50-50 is still 2,500, right?
Yeah.
That is at least 250 PhD students. Isn't it? So, like to find some candidates that are more likely to make heterodimers, if
Yes,
at all, right?
yes, I think that's doable, but need to find the 250 PhD students.
Right.